So just to review a little bit more about what genes are. So again, genes are the instructions that provide our bodies how to grow and function. We have over 20,000 genes in our bodies, and there can be changes or variations in our genes. Some are very common, and a lot of us in this room have minor variations in our DNA. It's part of what makes us unique. Some findings are what we call benign, and then other findings are what we call pathogenic or positive and cause the gene to not work properly. So those are referred to as mutations, and mutations are also referred to as pathogenic variants. So to further break down a gene, this picture up here shows that genes are composed of an exon and an intron. Exons are what are the protein coding parts of the gene. And they make up only 1 to 2% of the human genome, surprisingly, but they account for 85% of disease. The introns are the non-coding part of the gene, and they make up about 24%. So going back to this picture and thinking about as we transition into talking about genetic testing, this shows what you would want to test for whenever you're thinking about genetic testing. So if you're wanting to look at all the DNA, you would consider whole exome or whole genome sequencing. If you want to look for large missing or extra sections of DNA, you would think about a chromosomal microarray. And then if you've got a group of disorders or a condition that we know only is caused by one gene, you would think of a single gene or a multi-gene panel. So there is a current shift right now in genetic testing, and we're all kind of going toward whole exome and whole genome sequencing. That could be a whole presentation in and of itself. But specifically as it relates to epilepsy and the genetics of epilepsy, we know that epilepsy can be caused by structural brain abnormalities, trauma, infection, but 40% of individuals with epilepsy do have a genetic cause or a genetic etiology. Genes have been identified that cause both generalized seizures and focal seizures, as well as unclassified epilepsy, such as infantile spasms. The chance that a patient has a genetic cause for their epilepsy is higher in conditions like infantile spasms, Dravet syndrome, and generalized epilepsy. The inheritance can be dominant, dominant, recessive, or X-linked. And what we mean by that is if you go back thinking about the genes that we have in our bodies, we have 2 copies of every gene. One comes from mom and one comes from dad. If something is dominant, then only one of those copies is changed or altered. If something is recessive, then both copies of the gene are changed. And then if it's X-linked, that means that it was passed by on the X chromosome. De novo variants can also occur, and those just mean that they occur for the first time in the child. They're not inherited from either parent. The pathogenic or positive variants can occur in a single gene that may be associated with different types of seizures, and that's what we refer to as clinical heterogeneity. And then you can also have pathogenic variants in different genes that cause the same phenotype or same epilepsy, and that is referred to as genetic heterogeneity. Sorry for the slides got a little shifted, it looks like. So just going over genetic testing strategies, I think for some of you in the room, years ago, the recommendation was to undergo a chromosomal microarray, followed by possible a single gene or a multi-gene panel. Again, the array would look at large sections of DNA that was either missing or extra or duplicated. Single gene testing is for testing of when you have a specific condition in mind. And then multi-gene panels, so for instance, seizure panels or cardiovascular panels. I would say the challenge with panels is that they are outdated the day that they are released by labs. It's very hard to keep up with scientific data and have the most up-to-date panel. So that's why there is a shift to more recommend whole exome sequencing or whole genome sequencing. So look at all of the DNA. So if you want to think about this even more, we've got it broken down by the diagnostic rate by each testing option. So traditional panels only yield about a 10% diagnostic rate. A CMA is about a 10 to 20% diagnostic rate, whereas exome sequencing or even genome sequencing, it's anywhere from 31 to even as high as 60%. So your greatest diagnostic yield is going to be with a more broad approach. So as some of you in this room know, the benefits of genetic testing are numerous. It helps identify a diagnosis faster. It can end the diagnostic odyssey that patients often are on. You can develop a more precise care plan. It maximizes the individual's positive health outcomes. You can alert providers to additional health concerns that warrant screening, especially for those that find out they are have a syndromic form of a disease and have more than one body organ involved. You can avoid unnecessary follow-up testing and appointments. It's a way to connect parents and family members to specific support, hence how you guys are here today. Increase patient and caregiver satisfaction, and it can qualify patients for treatment options and clinical trials. So there was a recently, or I should say in 2021, the American College of Medical Genetics and Genomics, or ACMG, recommended as a first-line test whole exome sequencing for individuals with developmental delay, intellectual disability, and congenital anomalies. And then in 2022, the National Society of Genetic Counselors recommended exome testing as the first-line test for unexplained epilepsy. And then just this last September, the American Epilepsy Society endorsed that. So you can see that a lot of societies and individuals are moving toward this exome first approach to testing. So in a research study done by Smith et al., they found that 80% of patients with a genetic diagnosis had changes in their medical management, including avoiding, stopping, or initiating specific anti-seizure medications, starting a ketogenic diet, and halting a plan for surgery. So in order to kind of get to that best medical management plan quicker, exome testing is recommended. When we do exome testing, for those of you maybe in the room who have done that or have recommended it, we do recommend trio testing, and that just means having samples from each parent accompany the child sample. And the reason why we like to do this, because like I said earlier, all of us have minor variations in our DNA. It's part of what makes us unique. But by having a trio sample, we really reduce the possibility of what we call variants of uncertain significance, or AVUS, whenever we're analyzing the data and writing the reports. And so typically this does come from both biological parents, but when that's not an option, we can use siblings. So let's take a look at reports and break those down a little bit. So when you do genetic testing results, I used to always like to tell patients that there were 3 types of results that you can get. You can get a positive result, meaning we find an answer that currently matches what we know in the scientific literature to cause disease. We can get a negative result. And I always explain to families that a negative result does not mean that there's not something genetic going on in the family or in the child. It just means that as of current technologies, we have not discovered what that is. So it's often recommended that genetic testing is later reanalyzed and looked at again because of changes and updates in technology. And then the third type of result is a variant of uncertain significance. Like we said earlier, just means that there's been a change identified, but we don't know currently if that's causing disease or not. So these are obviously going to be reports that are generated by GeneDx and what these look like. And all of the reports that I'm going to show you in the next few slides are taken from patients. Obviously, they're de-identified, but taken from patients with a CHD2 mutation. So on the front page, you have your basic demographic information. You have information about the sample, if it was blood, if it was buccal, when it was collected, when it was received by the lab, and then when testing was actually started and then reported. You'll have the name of the test. So the test requested is the name of the test. So exome DX Plus means that an exome test was performed, and the provider who ordered the test will give some clinical indication terms about what the why he or she ordered the testing. In the next section of the report on page 1, you'll see here that whether the result is positive or negative will be in that blue box. And then it further breaks it down here with the bullet points. So causative variants in disease genes associated with the reported phenotype, you'll see that that's positive. And this next section is includes or encompasses a category that we use called candidate gene, where there's a change, could be a change in a gene that is highly suspicious of causing disease, but we're not quite sure of the classification. In this particular report, none were identified. ACMG secondary findings. So ACMG, again, stands for the American College of Medical Genetics. So anytime exome testing or genome testing is performed, the American College of Medical Genetics requests or has recommended that you test for 78 genes that are adult onset medically actionable disorders that can be found out from exome testing. And so when you're consenting for this test, parents or patients have to give their permission if they want this information released. And again, it's adult onset medically actionable, so heart conditions, cancer conditions. A lot of people get confused and think that this is like, you're gonna find out if you have dementia or if you have Huntington's disease, and that's not what these are. And then GeneDx also has the option of looking at mitochondrial DNA. And so for this particular report, the analysis was provided in a separate report. But also, again, these are just blown up sections of each report. This would still be on page 1. So you see here, again, we knew we had a positive report. So we have the gene that was found, so CHD2. And the next section includes the disease that it's associated with, the mode of inheritance, the variant. We're going to go over this in a few more slides, but I always explain to patients that's like the address and the gene of where the change occurred. So it's just like you and I have a home address. This is the address where this variant occurred. The zygosity, heterozygous, that just means there was one. If that said homozygous, there would be 2 changes. Inherited from this report, we know that this was a de novo or brand new change in the patient and not inherited from either parent. And then it's classified as pathogenic or positive. The interpretation section just provides kind of a brief overview of the gene and the condition that it's associated with. And then there's a list of recommendations for the ordering provider to consider next steps. Then starting at this section, you would get to page 2 of the report, and this goes into a little bit more detail about the gene, what disorders it's associated with, and what published articles have been written about the gene. Also, again, we'll restate that it was not inherited from either parent, and why and what's justified in classifying it as pathogenic. The additional analysis comment section just provides information about how the actual analysis occurred by the lab. ACMG secondary findings limitations. Again, that just goes over, like we said earlier, these secondary findings that can be looked at in testing. I'm not sure if I explained this earlier, but secondary findings are just that. They're not related to the primary reason for testing. And so that's how they get their name of secondary findings. And then you can have additional information on the report, including resources. So if there are any, what resources are available about the gene or the condition, we provide. And then if mom and dad submitted samples, we have that indicated on the report. The quality metrics is just about the actual sequencing reads for the data for their analysis. And then if there were any copy number variants detected, so copy number variants are those large deletions or duplications of genetic material. The method section goes over that, the methods, how the lab performed the test. There's a disclaimer in every report with every testing lab, no genetic testing is perfect. And then all the references that are cited that have been included that mention the gene or the disease associated with it. So just to go, in the last few slides, we're going to go over the actual types of variants that it can occur, specifically in CHD2 that cause disease. But before we do that, we're just going to go over a few basic concepts. So as we've already said, there's genes. Genes undergo a transcription process to create RNA. And then from RNA, you have translation to amino acids that then provide protein products that tell our bodies how to grow and function. And then this is a table of all the 20 amino acids in our bodies. We have what we call start codons and stop codons that are indicated in green and red at the bottom. And when genetic testing is reported, the variants are the variants that are named depict the change in the DNA, not the RNA. So DNA is a long stretches of letters. And then you can see here that you have a non-coding section of DNA, just like we talked about, that intronic region. And then you'll have a group of letters that actually code or make sense that then are translated into RNA and then into amino acids. So this is what it looks like in a healthy cell without any changes or variants that would cause any problems. And so this slide just goes over where specific changes can occur. So like I said earlier on from the test report, you can see that it provides an exact location or change in the gene that caused disease. And so at base number 136, a C was changed to a T. And then there also provides the exact codon region where this change occurred. And so that's denoted by the P. So at codon number 46, an arginine was changed to a stop codon. And on reports, if you see that asterisk, that represents a stop codon, and therefore diminished or decreased protein products is created. I know some of the talks today have mentioned these terms, and I always found it helpful to explain to parents, in a little bit more detail. So this is what we're going to use on the next few slides to talk about this concept. So like I said, again, you have the intronic region of genes that are just letters that don't make any sense. And then in the middle of all that, you're going to have a phrase that does make sense. Like for the purpose of this slide, the tan pot was hot. And then after that, you'll have another string of letters that does not make sense. So the and hot represent the start and stop codon. Each individual word represents the codon or the amino acid. And then the whole sentence represents the gene or the protein. There's a lot of words on the slide. And again, I apologize that they were, they're shifted a little bit. But these are the types of mutations that you may have heard your providers talk about, or maybe you've seen in your reports. So a missense variant is a point mutation where one letter or base is changed and one amino acid is different. And this sometimes can change, or it changes the meaning of the original sentence, and sometimes it can still make sense. And sometimes it can be illogical. So you can see the example here, the tan pot was hot. That was our original sentence. And then there was a change from a P to a C, so the tan cot was hot. And so you'll see that that can still make sense, but it's not the original protein. A nonsense mutation is a little bit different because there's one base letter that is changed and it ends the sentence too early, creating a stop codon. And then that usually degrades the protein that was supposed to be created. And then you can have what we call an end frame deletion, where within that frame, you have a deletion of one of the 3-letter words or codons. And again, it may still make sense, but then it might not make sense. It just depends. But the reading frame itself is not shifted. And then for an end frame insertion, it's just the opposite of a deletion. You have an insertion of a codon. And again, it may still make sense. But then again, it might not. And so it could affect the protein products. But again, the reading frame is not shifted. This just provides another picture of what a missense variant looks like, where you can see the top strand is what would be what we call a normal sequence. And then below that, you can see where in the middle, the A was changed to a T, a point mutation, but it changed the whole amino acid to a proline, and therefore changed the protein product or the original sequence of the gene. And this is what it would look like on a report if you were to look at this again. So under the variant column, you can see at base number 2095, the C was changed to a T. So again, a nonsense variant, it'll have that point mutation, but it results in a stop codon. Whereas you can see on that top picture, you should have more amino acids. And on the second picture, that one letter change creates a complete stop codon, causing an incorrect sequence and shortening the protein. And this is what this would look like in a report when you look at the actual variant. And again, you can see that the asterisk there under the variant section at codon 896 was changed to a stop codon. Again, there's a lot of letters on this slide, but frame shift is just that, it shifts the frame. So again, you have the non-coding section of that long strand of letters, and then in the middle of that, you have something that does make sense. But if you have a reading or a frame shift mutation, you can have an insertion of a letter that shifts everything after that insertion occurs. And so you can have a deletion or insertion, and it changes the letters that make up the codon, and then it'll change every codon after that part. And this could also lead to a premature stop codon. And again, this is just saying what we've already reviewed. It can be an insertion or deletion, but it changes the whole sequence of protein following where the point mutation occurred. And on these types of reports, you can see that under the variant section, you can see SS, and there's a frame shift that did result, because we have that asterisk there, it did result in a stop codon. The last type of variant that can occur that we're going to talk about is a splice site variant, and that is a point mutation that occurs at the boundary of an exon and an intron, and it can result in an in-frame insertion, a frame shift, or the whole exon being skipped. In this particular example, on the second line of the long letters, you can see that the A was supposed to be a G, but it became an A, and that resulted in this bottom box here of 4 additional amino acids added to the protein. This picture also just goes over types of splice site mutations. So in the top picture, it completely spliced out that intronic region. But on the bottom, the point mutation that occurred deleted the whole exon 10. And so then you had exon 9 and exon 11 join. And so for this, on a report that you would see that occurred as a result of a splice site variant, under the variant section, because it impacts the coding sequence, you'll see a question mark where it talks about where it would usually depict where what codon occurred in. All right, and I believe that is all. That was a lot of information.